Charlie Barr felt whenever it happened to him?
He was at Bellevue, wasn't he?
No, Charlie Barr was here.
Charlie was here. Laura was at Bellevue.
Correct.
She was there the year I was there.
So Charlie was like, he was a coach for 37 years. And Dale, whenever he retired, Dale decided to take away the freshman football coaching position from him before asking him. And he felt like that was a stab in the back. But then he later says that it was probably the best decision because he got to spend more time with some of the things that he's missed because of coaching.
Never easy.
Yeah.
Never, never, never. All right. All right, all right.
Thanks, man.
See ya. Hello.
Who's that?
So how do you want to do I need an iPad?
I don't think so.
So we can do the questions.
Oh, because it's too loud, right?
Typing.
I'll just keep it down. Oh, you want to do the iPad?
Well, I guess you could do it on your computer, but then we got to go back and forth between.
Okay.
Bianca, welcome, welcome.
Can you show you what it is?
Number 5 is about 10 centimeters.
Thanks, man.
About 10 centimeters. About 10 centimeters. Looks about. Huh? Um,
Okay, so there's probably 10.
All right, hey, welcome, guys. Happy 7th period on Thursday.
Sorry.
Hey, we are going to get started here in just a couple of minutes. It's like 1:45 right now. So let's shoot for starting right at 1:50. As you come in, make sure that you put your name and your teacher's name in the live chat so that we know that you're here, we know who you are. And then we'll start going through the PowerPoint and the discussion and the demonstrations and all that stuff as we get going. All right. Glad you guys are here. Let's get it going. Let's kick some butt. Let's make it happen. All right. So which one exactly one?
I would go ahead.
Okay.
That way. Yeah, you can just edit your stuff on there.
Awesome. Wednesday, make sure to have that piece of paper out.
Yep.
All right, all right. So Mr. Wen is putting a message into the live chat right now. We still have about 3 minutes until we get started. We're shooting for 1:50 for our start time. So make sure on your end that you guys have a piece of paper or a notebook or something that you can take down some notes, maybe draw some pictures, do some of that stuff as we're going through the lecture, just to kind of stay with it and do your part from home. So make sure you got something to write with, something to write on, and we'll get started here in 2 minutes, 2.5 minutes, something like that. All right.
Just let me know if you need anything.
Okay. I think we're good. I'm just gonna just keep an eye on the video. Make sure, like, if it gets jumpy or seems to be laggy, just let me know and we can kind of.
Well, if you wanna do it, let me know, will you jump in and jump out, how many times in 4 seconds?
Well, I think there's always going to be a little bit of lag. I'm not super worried about that right now. We'll see that when we get a little bit closer, or when we get into it, we'll be able to see what. So it's me and you, right? Your kids and my kids.
About, yeah, it's about the same amount of lag, probably.
Yeah.
Hey, Alex.
What's that?
Hey,
Alex?
Alex. Hey, Alex. Yes. Oh, what am I thinking? Alright, let's go.
Okay, guys, welcome to Thursday, 7th period biology. We're going to have a fun little live lecture here. We have Mr. Ewald's class, my class, and we also have Mr. Wen's class in the live lecture. So thank you for coming. Thanks for showing up for this. Again, make sure you put your name in the chat with your teacher's name. Make sure you have something to write with, something to write on. There's going to be some slides in here where we're going to be having you doing something as we go through. All right, so today we are going to talk about cell transport, so the movement of materials across the cell membrane. Before we really start getting into that new material, we're going to take just a couple slides to review what we looked at earlier, earlier in the week, about the cell membrane or the plasma membrane. Okay, so just to refresh your memory, on our first slide here we have a beautiful little picture of the cell membrane, the plasma membrane, and some basic information about the cell membrane. What we ultimately want you to remember, all right, is that the cell membrane is this thin, flexible boundary that separates the inside of the cell from the environment surrounding the cell. Every single cell on planet Earth has a cell membrane. Doesn't matter what it is, right? If it's a cell, it has a cell membrane. The primary function, all right, the main function of the cell membrane is to regulate the movement of materials into and out of the cell. So it's this wonderful little boundary. It's made of some different materials, which we're going to talk about here in just a second. But it's this wonderful little boundary that helps to determine what moves into the cell and out of the cell. So I'll give you 30 seconds, maybe a minute to jot that stuff down. and then we'll roll on to the next slide. All right, so let's roll on. Something very important when we think about the cell membrane being this boundary between the inside of the cell and the outside, and the major function being to determine what enters and exits the cell, this idea of selective permeability becomes very important. So selective permeability is just a fancy way of saying that the cell regulates or controls the movement of those substances into and out of the cell. To be permeable to something, that means that materials can move across the membrane from the inside of the cell to the outside or vice versa. And to be selectively permeable means that the cell membrane is what's doing, is choosing what is going in and what is coming out. So selective permeability, that is the ability for the cell membrane to control what enters and exits the cell. As we start talking about cell transport, the movement of specific materials into and out of the cell, we're going to see why selective permeability is so important. All right, so structurally, we have our cell membrane down here. We can see that the cell membrane is made of some different materials. First and foremost, the cell membrane is made out of what we call a phospholipid bilayer. Right? So when we look at this word, phospholipid bilayer, the cell membrane is made out of phospholipids. Right? These are special types of fats. And it's a bilayer. The prefix bi means two, so it is a double layer of phospholipids. Structurally, the reason that the phospholipids orient themselves as a double layer is because they are each composed of what we call a polar phosphate head. All right, so that's all these circular portions, these blue circular portions here and in here. So they have a polar phosphate head and they have a nonpolar fatty acid tail. So all of these little tails, these little kind of jellyfish-looking tentacles, are the nonpolar fatty acid tails. Okay? What I want you to think about, when we think about a cell, cells live in watery environments. So the cell membrane, the molecules, these phospholipids, set themselves up so that the polar heads are facing out and in, all right? Because they're polar, they want to interact with the water. And then our non-polar fatty acid tails are pointing away from the water in towards one another. So we create this wonderful, beautiful setup of a phospholipid bilayer, right? There are some other materials that make up the cell membrane, though, right? Which leads us on to our next slide, okay? The idea of the fluid mosaic model. So let's break this word down too. It's a model because it's a way of explaining the cell membrane. We say fluid because the cell membrane, the molecules that make up the cell membrane, actually move around or float around within the cell membrane. They're not connected to one another. All right? So the cell membrane is actually, the molecules that make up the cell membrane are actually going to be able to move around within the outside and the inside, and that allows it to behave more like a fluid. Okay? It's not tightly packed together. The materials can actually move around like a fluid. All right? So the fluid portion of the fluid mosaic model says that. Those materials can move around within the cell membrane. The mosaic portion means that the cell membrane is made up of a whole bunch of different parts, different pieces. We have our phospholipids, right? The blue and the tails, the blue and the tails. We also have very important membrane proteins. Okay, so these orange pieces that we have in here, okay, These are all of our membrane proteins, and we'll see a little bit more about those here in the next couple of slides. We also have cholesterol. Okay, so in our picture, we can see some of these little cholesterol molecules. They're kind of hard to see. We don't have a whole bunch of them on here. But there are cholesterol molecules in the cell membrane. And if you remember from earlier in the week, cholesterol helps to maintain the fluidity of the cell membrane to keep it from becoming too tightly packed or too separate. So we have cholesterol, and then we also have these carbohydrate chains that stick off some different types of proteins and phospholipids on the cell membrane. All of these materials, proteins, cholesterol, carbohydrates, phospholipids, they all work together to make this fluid mosaic model for the cell membrane. All important, all have important jobs for the function of the cell membrane. All right? Give you a second to jot that down. I know there's quite a bit on the slide, and I was probably standing in front of it. So I'll give you a couple of minutes to jot that down. Well, minute, maybe 2 minutes to jot that down, and then we'll roll on. Also, let me know if I seem to be going too fast or you're struggling to get the stuff written down before I jump from slide to slide. Make sure you put that in the live chat so that I know to kind of slow everything down and give you the time that you need to get the information down on your end. All right. So we have our cell membrane, we have our plasma membrane, we have our phospholipid bilayer. Okay? We understand that there are all of these different materials that are going to work together to allow the cell membrane to regulate what is moving across it into the cell or out of the cell or whatever that might be. Okay? So now we're going to kind of transition into looking at different types of cell transport, the way that materials move across the cell membrane. So the first thing that we're going to look at, the focus of our discussion today, is the idea of passive transport. Okay? So let's kind of look at a definition for passive transport before we move on any further. So passive transport, and I'm going to point out the most important part of this definition. So passive transport is the movement of particles or the movement of materials across the cell membrane without using energy. Okay? So on your end, whatever you're doing, underlining, highlighting, circling, putting a star next to something, without using energy is incredibly important. So when we talk about these different types of passive transport, what we always want to keep in the back of our minds is the fact that all of this is happening without the cell needing to use any energy. Okay? They're naturally going to happen without the need for any energy from the cell. Okay? The different types of passive transport that we're going to talk about today: diffusion or simple diffusion, facilitated diffusion, and osmosis. So we're going to provide some definitions. We're going to give you some examples. We're also going to do some really kind of quick, simple little demonstrations to show you how that's going to work. All right? So we have passive transport, the movement of particles across the cell membrane without the need for energy. That's big. Diffusion, facilitated diffusion, and osmosis. So let's dive in. The first type of passive transport that we're going to talk about is diffusion. Okay? So diffusion is the movement of particles or materials that are dissolved in water from an area of high concentration to an area of low concentration. On the last slide, I had you underline, circle, highlight, star, whatever, the phrase without using energy. So on this slide, I want you to pay special attention to from an area of high concentration to an area of lower concentration. Okay? So materials, particles, are going to diffuse across the cell membrane without using energy from an area of high concentration to an area of low concentration. The best way of thinking about this is you walk down to the Ohio River, you got yourself a canoe or a kayak, you throw the canoe in the Ohio River, you jump in, and you allow the current of the river to take you where it's going to take you. You can basically just sit in that canoe and let the current take you. All right? Doesn't require much, if any, energy from you, and you're just moving with the current of the river. That's exactly what's happening with diffusion. Materials are moving from high concentration to low concentration without energy. So we have a wonderful little picture here that's going to show this. At time 0, we have a beaker that has some water in it, and over on this side we have some red dye or molecules. On this side we have some blue dye or molecules. After 5 minutes, we're going to see how those molecules are starting to move from areas of higher concentration and spread out within that water. After 10 minutes, we're going to see that those molecules have really spread out within that solution. Maybe water, maybe something else, but we see how those molecules are going to diffuse. Okay? A very simple, actually, before I do that, I'm going to do a quick little demonstration here. And I got 4 colors. So I'm going to let you guys choose. So everybody that's on the live lecture right now, we have red, we have yellow, we have blue, and we have green. So red, yellow, blue, or green. Pick a color. Everybody in the chat, go ahead and put down which color you want me to use for this demonstration. Go ahead. I'll give you 30 seconds to put that in the live chat. Mr. Wen, let me know when those start coming in. We'll see what colors we like. Or color.
We're getting some blue. 1 red, 1 green, mostly blue. Ooh, 1 yellow. Yeah, I think it's blue. We're gonna go with Highlands blue or Memphis blue.
Or UK blue. So I will tell you right now, this is what I was originally going to go with, blue. Highlands blue, UK blue. Mr. Wen has his background with Memphis blue. But we're going to go with blue. Whoever you support is going to be their color, right? So I have a simple cup of water right here, right? Nothing crazy, just plain old water. I got my blue food coloring. So this is something that you can do at home, and I'm sure you've seen this at some point, but never really thought about the fact that this was showing diffusion. So we have our cup of water here, and I'm basically just going to put a couple drops of food coloring in there, and what you can see is that as time goes on, and we're talking about just a series of seconds, we're going to see how that blue food coloring is going to want to diffuse from an area of high concentration to an area of lower concentration. So the pigment, the molecules, the material that makes up that food coloring is going to want to spread out amongst those water molecules. Okay? Now, we can do something. We can apply a force like temperature or movement or something like that to make this happen quicker. Okay? So I can actually spin the cup, and let me try to do this without spilling it all over Mr. Wen's stuff. Okay, so I can mix the cup around, I can mix the water, and you can see how that helps the food coloring diffuse throughout a little bit more. Still a little bit of dark at the bottom, but you can see how it's starting to diffuse into that cup. Okay, so a simple little demonstration to show you how materials naturally, without energy, want to move from an area of higher concentration to an area of lower concentration. I'll let this sit here just on the desk, and maybe if I remember, we'll check back in a little while and see what it looks like later on. Okay, so that's diffusion, simple diffusion. All right. Now, over here we said that materials want to move from an area of high concentration to an area of low concentration. Why do they want to do that? They want to do that because they are trying to reach what we call dynamic equilibrium. Okay? Dynamic equilibrium is reached when the diffusion of material into or out of a cell equals the diffusion of material into or out of a cell. So molecules will continue to move, but the overall concentration remains the same. So the materials are going to want to move from high to low until the concentrations are equal. So over here we have a very simple picture with some dots, and we have a cell membrane. These dots are going to continue to move until they reach dynamic equilibrium, until they reach a situation where the concentration on this side is the same as the concentration on that side. Same thing is true with our food coloring and water. So the molecules will continue to diffuse within this cup until they are evenly spaced throughout the entire cup of water. All right? Dynamic equilibrium. Okay. So that brings us to our first question. So on your end, what I want you guys to do is go ahead and draw this on your paper. Okay? So we have a setup here. We have a membrane in the middle, and we have 6 circles or 6 particles on the left-hand side. So what I want you guys to write down on your paper is drawing arrows, show the movement of the materials, and then do a quick little explanation of what would happen over time for this cell to reach equilibrium. All right? So let's get the live chat going here a little bit. If you are in Mr. Ewald's class, and your last name is between K and R, so Mr. Ewald's class, K and R, go ahead and throw into the live chat your explanation of what would happen over time for this cell to reach dynamic equilibrium. All right, give you just a couple of minutes to get that drawn down and to answer that question, and we'll come back together.
Yeah, Memphis Tigers, but the Memphis Blues, I think, might have been like their hockey team. But there are the River Kings as well. So the Memphis River Kings.
Gotcha. All right, so again, Mr. Ewald's class, K through R. Go ahead and put your description of how you think these materials, these molecules are going to move in order to reach equilibrium.
All right, we have Alex that has said there will be 3 dots on both sides. Caroline, there will be the same amount of dots on either side.
All right, so thank you to those of you that put your answers in there. You're absolutely right, okay? The materials will diffuse across the membrane until they reach a state of equilibrium. So ultimately what we're going to have is 1, 2, 3 particles are going to diffuse until we reach equilibrium, right? This is not there anymore, that's not there anymore, that's not there anymore. So we have 3 on this side and we have 3 on that side. Wonderful. All right. Now, let's jump into this one, okay? So we have a very similar image, okay? We have our membrane in the middle, we have 2 sides. Now we have some materials on the left side and none on the right side. So what I want you to think about here is, again, using arrows on your paper to show what's going to happen. What would happen over time for this cell to reach equilibrium? If you are in Mr. Wen's class, everybody? All right, let's do everybody. Okay? Mr. Ewald's, Mr. Wen's, everybody, go ahead and put your answer in the chat. What do you think is going to happen with this picture in order for the cell to reach equilibrium? Do your thing.
All right, we got two particles we need to travel to either to the other side. Two of the dots move from the left to right to achieve equilibrium. Two on each side. 2 and 2, 2 and 2, 2 and 2.
All right, so it seems like the prevailing answer is that two of these dots are going to move to the other side to reach equilibrium. So what I basically am going to do is I'm going to take this dot, I'm going to draw it over here, and I'm going to take this dot and I'm going to draw it over there. Is that right? Is that, in order to reach equilibrium, that's what's going to happen?
No, Mr. Ewald, you can't do that.
I can't do that.
Well, look at the size of the dots.
Okay, they're definitely bigger.
And they don't fit the hole in the membrane.
Oh, so we have a little bit of an issue here. Is that what you're telling me?
You're absolutely right.
These dots are too big to fit through the hole in the cell membrane. So we couldn't actually have this. We're just going to have these four staying on this side and none on that side.
That is correct.
Would you agree with that?
Absolutely.
So we have a bit of a problem, okay, that we actually have a solution for, so stay with me. The problem here is that these particles are too big to just move passively across the cell membrane without any help, okay? This leads us to the solution, okay? The solution is, I'm going to skip that one, I'll come back to it. The solution is facilitated diffusion. All right, so our next type of passive transport, which is facilitated diffusion, and that is the movement of materials across the plasma membrane using proteins. Okay? So in this case, the most important part of this is this idea of using proteins. So it's the movement of materials across the plasma membrane, the cell membrane, using proteins. So what we see in these two pictures, we have our phospholipid bilayer. We can see that here, we can see that here, here, and here. But what we now have sandwiched within the plasma membrane are membrane proteins, channel proteins, carrier proteins. So when we have materials that are too big to simply move across the cell membrane, or when we have materials that are charged, polar materials, we need something to help those particles move across the cell membrane. And what is it that helps that? It's proteins. So the process of facilitated diffusion is the movement of these materials across the cell membrane using proteins. So what we can see in this picture, we have green molecules and we have the big red molecules. The green molecules are small enough. They can move directly across the cell membrane using diffusion. But the big red molecules, they're going to need a protein to help them get from one side to the other. The materials are still going to move without energy, and they're still going to move from an area of high concentration to an area of low concentration. They're just going to use a protein. Okay? So let's jump into this really quickly. Thinking about simple diffusion and facilitated diffusion. Okay? What are two differences between facilitated diffusion and simple diffusion? And what are two similarities between facilitated diffusion and simple diffusion? So we're going to do a little similarities and differences between simple diffusion and facilitated diffusion. All right, so on your end, on your paper, go ahead and write down two differences and two similarities. In the live chat, because we have so few students in both of our classes, I'm going to open it up to everybody. Okay? What I want you to do is go ahead and put down one of your differences and one of your similarities in the live chat. I'm going to give you a couple of minutes to jot all that stuff down, and then we'll come back together and we'll go from there. All right? Two similarities, two differences on your paper, one similarity and one difference in the live chat. Let's do it. All right, so again, if you could, in the live chat, go ahead and put down one difference between facilitated diffusion and simple diffusion and one similarity between facilitated diffusion and simple diffusion.
Alrighty, we
What do we
have
got?
a difference being that facilitated diffusion uses proteins while simple diffusion does not.
All right?
A similarity, both involve substances going from an area of high concentration to an area of low concentration.
Love it.
Okay? So, yep, another student said the same thing. Uses proteins and simple does not. They both go from high to low.
Beautiful. All right, so let's kind of think about this. Take what we got from the live chat and kind of put it in to this slide right here. So two major differences between facilitated diffusion and simple diffusion. Number one, facilitated diffusion requires a protein for the movement of materials, while simple diffusion does not. Facilitated diffusion is the movement of larger, charged, or polar molecules, while simple diffusion is the movement of much smaller, right, uncharged materials directly across the cell membrane. So those are two major differences between facilitated diffusion and simple diffusion. Similarities, both are passive. So both, there's no energy that's needed. Okay? Another similarity is that both move materials from high concentration to low concentration. Because they're passive, we know that they're not going to use energy, and they're both going to move materials from high concentration to low concentration. So wonderful job, great job there. I just noticed, because we're getting ready to wrap up diffusion and facilitated diffusion, here's my cup that I put the blue food coloring in earlier. What you can see now is we've had diffusion within that cup. All right, so those food coloring molecules have evenly spread out among the water molecules, and now we have this nice kind of even blue color in the cup. All right. So, let's move on. Oh, we got one more picture. Sorry, question number 4. On your paper, go ahead and draw this down, right? We got our membrane, right? We now have something in the membrane, so you're going to use arrows and you're going to draw and explain what would happen over time for this cell to reach equilibrium. Give you a minute or two to jot that down, and then we'll go from there. I'm not going to put it in the live chat. No, there's one. I'm just going to share it. Since we have so few, it's like the Tennessee ones. I'll have them. I'll have them here. All right, so let's kind of wrap this up. So what we have here is a picture, right? We have 4 particles on the left-hand side, none on the right. What we now have is a membrane protein. So we've created the bridge from one side of the cell membrane to the other. So just like we thought was going to happen earlier on that wasn't able to happen, we're now going to have 2 of these particles move to the other side so that the cell is now at equilibrium. Okay? So this is a perfect little example of facilitated diffusion. All right, good stuff, guys. Hopefully you got that on your end. I know I didn't have you put anything in the live chat, but we're just going to roll on. All right, so here we go. Our last type of passive transport is something that we call osmosis. Okay? Osmosis is the diffusion of water across a selectively permeable membrane, across the cell membrane. It is passive transport. So the materials are going to move from an area of high concentration to a low concentration until they reach equilibrium. So again, osmosis is special because it deals with the movement of water. Diffusion and facilitated diffusion deal with the movement of particles that are dissolved in water, while osmosis is going to deal with the diffusion of water. There are 3 different types of solutions that we're going to look at as we think about osmosis. The first is what we call an isotonic solution. The second is what we call a hypotonic solution. And the third is what we call a hypertonic solution. Now, these may seem kind of scary right off the bat. They may kind of seem like they're very similar. Okay? So what we're going to do is we're going to look at the prefixes of these words to see if that can help us out a little bit. All right? So the first is isotonic. So we want to look at the prefix iso. Okay? So if we think of this prefix iso, so if we're thinking about an isometric exercise, all right? If we're thinking about any other word that starts with iso, okay? What iso means is same. Okay, so what we're going to see here in just a minute is something about an isotonic solution is the same. All right, hypo, all right, the first word that probably comes to mind is hypothermia. So if we think of somebody that is hypothermic, that means that their internal body temperature has reached a level that is incredibly below the normal body temperature. Okay, so the prefix hypo means below. And then hyper, all right? The first word that comes to mind in my mind is hyperactive. Okay, so somebody that is hyperactive is going to have an incredibly high level of energy, way above the normal level of energy for anybody else. Okay, so hyper means above. Okay? Iso means same, hypo means below, and hyper means above. So we're going to take a little bit of time here over the next couple of minutes to look at how these words play a role in our understanding of osmosis. Okay? So let's start with isotonic. So in an isotonic solution, what we are going to see is that water and dissolved substances are going to diffuse into and out of a cell at the same rate. So the amount of water that's entering the cell is the same as the amount of water that's exiting the cell. So we're not going to see a change in the size or volume of a cell overall. So what we have here is a nice little picture of an animal cell. All right. What we can see is that the concentration of orange solutes, dissolved particles in water, is going to be the same as the concentration of dissolved particles outside of the cell. All right. Therefore, the concentrations of water are going to be the same. So we're going to see some water leaving, and we're going to see some water entering. Okay? But the overall volume, the overall mass, the overall size of the cell is not going to change. Okay? Remember, iso means same, so the concentrations are going to be the same inside versus out. All right, so let's look at a hypotonic solution. Okay? So with a hypotonic solution, what we're going to see is the concentration of solutes is going to be higher inside of the cell, but lower or below on the outside. Okay? So in a hypotonic solution, the solute concentration is higher inside the cell versus outside. So what's going to happen is water is going to diffuse via osmosis into the cell. Okay? So water is going to diffuse through osmosis into the cell, so the cell is going to swell up. The volume is going to get larger. Potentially the mass of that cell is going to get larger as the water enters the cell. Something to remember, and this is super important. So I'm going to put a star next to it. Okay? Water will always move or go towards where there is more solute. All right? So in our hypotonic solution, there is more solute inside of the cell and less outside, so the water is going to want to move in. Water is going to move into the cell is going to swell up and potentially get larger in volume. Okay? So again, in a hypotonic solution, water is going to move into the cell, and the cell is going to swell up. Why? Because the water is moving towards where there's more dissolved solutes. Hypotonic. All right, and our last type of solution is hypertonic. Okay? So in a hypertonic solution, the concentration of solutes is now going to be higher outside of the cell. Remember that hyper means above. So the concentration of solutes is super high outside of the cell. All right? So what's going to happen then? Water is going to want to move towards where there's more solutes. So there's less solutes inside, more solutes outside, so the water is going to want to move out. All right? That means that potentially the cell will shrivel up. It's going to lose water. It's going to lose volume. based on the fact that the water wants to move out towards the more solutes. Okay? All right, so we had isotonic, we had hypotonic, and now we have hypertonic. So based on the concentrations of dissolved solutes, based on the concentrations of water, we're going to see water moving potentially in some different directions. So let's do some practice. So for question number 5, we have this wonderful little red blood cell that's sitting in a solution. Okay, so what I want you to do on your end is draw this picture, and using arrows, I want you to predict the movement of water. What I want you to pay attention to are the percentages. So the percentage of solute, the percentage of water, the percentage of solute, and the percentage of water. Okay? What I also want you to think about is what kind of solution do we have based on the movement of water. So everybody, both Mr. Ewald and Mr. Wen's group, in the live chat, once you've had a chance to answer this on your own, go ahead and throw your answer in there of which direction will the water move and what kind of solution do we have? Hypertonic, hypotonic, or isotonic? Give you a couple of minutes to do that, and then we'll come back together. All right, Mr. Wen, let me know when we get some answers in there and we'll solve this bad boy. So we got a red blood cell. We have some differences in concentration. Where do you guys think the water is going to move based on those concentrations?
Water will move from the inside of the cell to the outside of the cell because there's more solute outside the cell. This is a hypertonic solution.
All right.
Hypertonic because the water is going to move to the outside of the cell because there is a higher solution. Move to the outside of the cell, hypertonic, 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 moving outside.
All right, nicely done, guys. So if we look at the concentration of solute, we see that there is 75% on the outside, 45% on the inside. We know that water wants to move towards the higher concentration of solutes. So water is absolutely going to move out of the cell. And when we have a solution that causes water to move out of the cell, we have a hypertonic solution. So wonderful, wonderful, wonderful job with that one. Okay? Let's look at another example. Question number 6. Same thing. We have some concentrations on the outside of water and solutes. We have some concentrations on the inside water and solutes. We have a red blood cell in a solution. On your paper, draw down using arrows, right? Which way is the water going to move, and what kind of solution do we have? Mr. Wen's class. All right, Mr. Wen's class only. Mr. Ewald's class, you get a little pass on this one. Mr. Wen's class in the live chat. Go ahead and write down your prediction of where the water will move and what kind of solution we have. Give you a couple minutes and then we'll come back. All right, Mr. Wen, let me know if we have any answers.
Alrighty, so hypertonic, water will move into the cell. It's hypotonic because water is going to move in the cell. There's a greater concentration. Hypotonic, there is more solute in the cell.
All right, good answers, guys. Wonderful job. So if we look at our solute concentration, we have 45% inside, 40% outside. So we have a higher concentration of solutes inside the cell. Water is going to want to move towards the higher concentration of solute. That means that we do, in fact, have a hypotonic solution. All right, wonderful, wonderful, wonderful job there. Okay, as you can probably guess, we're going to do one more question, number 7. For the sake of time, we're going to do this one together, but I still want you to go ahead and write it down in your notes. Okay, so we have our red blood cell, we have our solution, and we have our concentrations. Okay, so what I want you to think about, I'm going to solve it here in just a second. What I want you to think about is where will the water move? So show that using arrows, and then tell me and Mr. Wen what kind of solution we would have. I'll give you a minute or two to solve that one, and then we'll do it together. Feel free to throw an answer in the live chat if you want to, right? If you want to get involved and put that answer in there in the live chat, you can do that. We'll solve it here in just a second. All right, so here's what we have. With our last question here, question number 7, we're going to look at the solute concentrations. we have 45% outside and 45% inside. So the solute concentration outside and inside are actually the same. What that means is that we are going to have some water moving towards the outside, but we are also going to have some water moving towards the inside. All right? The amount of water moving outside cancels out the amount of water moving inside, so we don't see a major change in the volume of the cell, and that's going to be our isotonic solution. Hopefully you guys got that on your end. So we've seen hypertonic, hypotonic, and isotonic examples of each of those cells. All right. Let's wrap up with this. Okay? So for our last question, we have one of my most favorite demonstrations or labs that we can do dealing with the movement of materials into or out of a cell. So what I want you to do really quickly is go ahead and jot down this simple table in your notes on your end. Okay? What we have done is we have taken good old-fashioned eggs that Mr. Nguyen bought at the grocery store, okay? And we put those eggs in some different solutions, all right? So we took some eggs. Mr. Nguyen took the mass of the eggs before, and then we put them in some solutions. So we put an egg in water, and we put an egg in Karo syrup, which is basically a really thick, sugary syrup solution. Okay? We left them in there for 24 hours. And what we want to check to see is what happened to the eggs, what happened to the mass of the eggs as a result of being in those solutions for 24 hours. So our initial mass for the egg in water was 66.5, and our initial mass for the egg in Karo syrup was 62.3 grams. So I'm going to take you off the stand here, and I'm going to move you over to our experimental setup and show you what we got. All right, so down here we have our digital scale, all right, and we have our two eggs. So the egg right here, which you can see, right, kind of squishy, looks like that. This is the egg that was in water. And over here we have our egg, and this is my favorite egg. This is the egg that was in Karo syrup. What you can see is that it's very squishy, and it looks like this. Okay, so in your table, what I want you to go ahead and do is, I'm going to take the mass of the egg after 24 hours that was in the water. What you can see is that the final mass of that egg is 84.2 grams. It started at 66.5, and it's now at 84.2 grams. So go ahead and jot that down in your table. 84.2 grams. All right. Our second egg, which is right here, this was the egg that was placed in Karo syrup for 24 hours. If we take the mass of this egg, what we now have is 44.43 grams. So we started at 62.3, and now we're at 44.43 grams. So what we can actually see is that based on the solution that we put the eggs in, this egg was water, and this egg was Karo syrup, you can see that materials absolutely moved into or out of the egg based on the concentrations of water and solutes in the solution. Awesome. You can do this on your own at home. Get some eggs, get some vinegar, get some different solutions, and see if you can modify the size or mass of the egg based on the solutions that you put it in. Super duper cool. so let's wrap this up. Thank you guys for being good sports and following along with us as we went through it. All right. Nice little review of the cell membrane, the structure of the cell membrane, the function of the cell membrane, and some great information about passive transport. So what I want to leave you with is, okay, make sure before you finish up with 7th period today that you turn in your questions by the end of the day. Okay? Check your Schoology page for instructions on how to submit. I know that Mr. Wen has something on his Schoology page. I absolutely have something in our Unit 3 materials on our Schoology page. Make sure that you go ahead and do that and submit it. Okay? Also check Schoology to see whether your teacher has assigned any more work. All right? So check Schoology and see if there's any more work in your Schoology materials for passive transport. And then our next time-bound event will be a small group or individually. Actually, on Monday and Tuesday, we are going to have our next lecture on active transport. So for you guys, that's going to be, I think, Monday. So on Monday, we're going to meet again via YouTube Live for a lecture on active transport. All right? Thank you, thank you. Enjoy the rest of your day. Make sure you get onto Schoology and take care of those assignments. And we'll see you guys on Monday. We'll see you on Monday. All right? Have a good one, guys.
All right, so just real quick, I know this is probably the first time you guys saw me, so I was behind the computer the whole time typing. Just make sure to check on Schoology, just exactly what Mr. Ewald said. There is an additional assignment that you want to make sure to do, and that's going to be due on Friday, okay? So it was good seeing you guys. I will see you guys again on our next live lecture.